So I think everybody's used to hearing about chemotherapy and other drugs, and it's what I'm going to talk to you about is really making the news, and is changing totally the environment of therapy. We used to have chemotherapy and surgery and radiation therapy, and the big, I think the big advance was getting monoclonal antibodies. And that was more than 10 years ago already. But we haven't had such a great breakthrough in the last 20 years as the use of cells. And it is the ability of cells to actually be able to target, to link with the cancer, and actually kill the cancer cell. And although a number of journals are reporting on boost your immune system and use these vitamins or whatever, which are usually not working, or there is no data to support any of these assertions. Clearly cells are doing a wonderful job, and we're only seeing the beginning of what they can be doing. So interestingly, if we want to start, like, why did we look at cells to be able to kill cancer? And this all stems from allogeneic hematopoietic cell transplantation. And it's clearly one of the big medical breakthroughs of the last 50 years. And people don't recognize, but this can cure approximately half of the patients who have refractory hematologic cancers, like blood cancers. And I'm talking acute leukemia, chronic leukemia, lymphoma, myeloma, which are not amenable to regular therapy. And by far, it's the most effective form of cancer immunotherapy right now. And cure by allogeneic transplant is what we call graft versus tumor effect, GVTE, or graft versus leukemia activity. And it's mediated by donor T cells that react against, for example, minor histocompatibility antigens or other antigens that are present on cancer cell. And what I would like to talk to you about is how can we take advantage of such activity. And I'm going to spend some time discussing various approaches related to stem cell transplant. So for those of you that are not doing transplant on a daily basis, I thought I would explain a little bit how it works. So if you have a patient with cancer, and the cancer is schematized by the yellow cells that you see in this patient, and you have his blood. If you look into the blood, you have red blood cells, and you have white blood cells, and you have platelets. Now, when these patients fail regular therapy, like chemotherapy or radiation or antibodies, we take them to stem cell transplant. And the idea is to give high doses of chemotherapy and radiation to eliminate the cancer cells. However, when we are doing that, we are also killing the normal bone marrow, and therefore patients rely on transfusions for red cells and for platelet. But you can understand that this is not able to totally compensate for the loss of stem cells, and there is a need clearly for replacing these cells. If we waited long enough, probably these patients would recover normal marrow, but it would take months before they do that, and then the patients would die of infection. So where do we replace these cells? What are the sources of cells? Well, usually we can go and try to look in the family if there's a donor that is HLA compatible. And we discussed that this afternoon. There's about 1 person in 4 that has a donor in the family that can be used for a transplant. Now, this leaves 75% of patients without a familial donor. So therefore, we go and look at what we call the unrelated registry. So we look worldwide into a bank of people that have said, I'm ready to donate my cells if I'm compatible with somebody else, with somebody who needs that, leukemia patients, for example. And with this unrelated registry, which is now more than 20 million people around the planet, we are still not able to find donors for everyone. And still between 30 and 45% of the patients don't have a donor. So therefore, for these patients, for which we didn't have a transplant not long ago, Now we have two options, either using umbilical cord blood transplant, which is cells that are normally discarded. They're taken at the time of delivery. The cells are being simply taken from the peripheral blood, and they're stored in various banks around the planet. They're typed and then available for donors. The other option is to look in the family if there's somebody that's what we call haploidentical. So half match, not a full match like a family or unrelated donor, but a half match individual that for which we could do a transplant, and I'm going to talk a little bit about that in the next slides. The other option is to use the patient's own cells and to freeze them before doing the transplant. But as you can imagine, if the patient has infiltration of his bone marrow by the cancer cell, then if we would be freezing not only stem cells but also malignant cells, and therefore there is a risk of reintroducing the malignant cells after the transplant. So we will stick with this opportunity for most of the patients with acute leukemia and a number of hematologic disorders. And what we do is then we take the stem cells. Most of the time we now use peripheral blood. We don't go to the operating room anymore. We give patients or donors, we give them growth factors and simply collect the cells a few days later in what we call an apheresis, basically filtering blood to collect the stem cells. So we take these cells and, sorry, I was pushing the wrong button, and reintroducing so that therefore we are able to regenerate a hematopoietic system with red blood cell, white blood cells, and platelets. But then you said the title is on immune therapy. Why am I showing you this? Because actually the cells I'm claiming are replacing the hematopoietic system, but they are also realizing another major task. And that a lot of the time the chemotherapy or the radiation is not sufficient to eliminate all the cancer cells. And actually I cheated a little bit because normally there are some leukemia cells in there. And we're counting on the cells that coming from the graft to actually fight out the residual cells. So I told you that we would be talking about haplotransplant. So I apologize for the students that were here this afternoon. Some of that we already discussed, but the rest is other things. So when we have somebody like a parent, so when we call haploidentical is somebody that for which the transplantation antigens are half matched, so a haplotype. So you get the white blood cell group on chromosome 6 and you're able to transfer that to your progeny. So actually if you have a parent here and he's giving his cells to his son or his daughter, we already know that they are 50% identical, and the other 50% coming from the mother, for example, in this specific example. So when we are taking the parent's cells and administering them to the patient in order to reconstitute an immune system, these cells can recognize various tissues. They can recognize the liver, the bowel, the lungs, the eyes, for example, the skin, and actually any organ. But these cells also have another function, and they can recognize infections like viruses or bacteria here, fungi, and actually leukemia, which is really crucial to what we're talking about. Have we been doing haplotransplant for a long time? We tried that like 30, 40 years ago. And when we did transplant with these individuals, patients all died. They all died because of this reaction of recognizing normal tissues. In addition, there's a lot of graft rejection. So because they're not identical, even the body, although you're giving high dose of chemotherapy and radiation, they're still able to reject the marrow that is coming in from somebody that's not perfectly matched. So what would we like to do? So what we would like to do is to be able to eliminate those cells that cause the deleterious effect, but actually preserve cells with anti-infection and anti-leukemia activity. Is it possible to do this? Well, we developed a strategy where we are taking donor cells and exposing donor cells to patient cells, and therefore doing exactly the same reaction that occurs in the patient, but we do that in the laboratory. So we take cells from the peripheral blood of the donor and mix them with cells from the peripheral blood of the patient in the lab. After 4 days, the cells of the donor that are able to recognize the patient, these cells become activated. So actually they turn red. And when we expose these cells to a dye, which is called TH9402, a rhodamine-derived medication, actually this medication accumulates in the cells that are activated, but those other cells that are able to recognize the infection and recognize the leukemia are not seeing the appropriate antigen, and therefore these cells are resting and they do not internalize the dye. And we have identified the mechanism, and because these cells internalize the dye basically because they're not able to pump out this medication. Since they're busy, their system is busy with the activation and the recognition of the antigens, this pump is actually inactivated, and then the dye is sequestered inside the cell. Now when we expose the cells to light, they're eliminated because this agent is a photosensitizer and releases oxygen radicals and therefore kills these cells. And therefore we can use this to do transplant to patients in need of a stem cell transplant. Very nice in theory. Does it work? I guess that was your next question. So we went into a clinical trial. And we said, let's test this strategy. Let's give these patients high doses of chemotherapy and radiation and give them what we call CD34 selected cell grafts. So stem cells, which are actually express this antigen, CD34, that is immunomagnetically separated. So we can get the purified samples of these cells and therefore eliminate the immune system so that we can get engraftment. We can get red cells, white cells, and platelets, but no immune system. And a month later, we administer what we call a donor lymphocyte infusion, a DLI. And we do that with increasing doses of the T cells. And we started with 1 × 10⁴, so 10,000 cells per kilogram, all the way to 5 million cells per kilogram. Now you have to know that people have done this part of the study. So people in Italy and in Israel have actually developed this protocol on which we have built, and they have administered that to patients. And they found that if this graft contains more than contains 3 × 10⁴ cells per kilogram or more, it is lethal. This small amount of cell is sufficient to recognize the patient and kill him. So that's why we chose a number to start with that was just below this threshold, but we actually went all the way 200 times above that threshold. And what we found is that the patients that received low doses of these cells, they had a survival that was not great, and you have to recognize that these are all patients with very bad diseases. These are all worst case scenarios, patients that failed chemotherapy, that had failed everything. So we had 20–30% survival, or somewhat less, 15 to 20%. But interestingly, there was a cohort in which we have had very interesting results, and the cohorts 4 to 6, those that were receiving higher numbers of these cells, of these immune cells, actually their survival was in the 60 to 70% range. Interestingly, these patients had low numbers of infection, low relapse rates. But if we went higher, and at the highest cohort, then what happened is we started to see some graft-versus-host disease, and actually the outcome was not any better. So we thought that we were able to identify a number of cells that we could administer to patients that were able to fight infection and fight leukemia. But then you wonder, is that true? Like you did one study, it was 19 patients, small group. Well, we went internationally, and a number of groups in Europe and in Canada. We did 23 patients at that dose. And interestingly, in what we call a phase 2 a study, which is there to evaluate the efficacy of the process, when we looked at graft-versus-host disease, so this reaction of donor cells against the patient, there was no severe graft-versus-host disease, no grade 3 or 4, which is a limiting toxicity for this treatment, and very few cases of graft-versus-host disease. And you can see here the incidence. So you can see at 1 year, only 20% of grade 2 GVHD, which is well controlled, and interestingly could be controlled with medication usually lasting only 6 to 8 months, while there was no grade 3 or 4 GVHD, and importantly, where we had very low relapse rates. So this is very encouraging. This is particularly encouraging when we compare that to what we had before, because I told you that other groups were able to do the same type of transplant, but without adding these cells, the immune cells, which were deprived of those that recognize the patient. So what other groups had been seeing, and actually is the same as what our group of investigators had seen when we did exactly the same treatment. So the difference between the two curves is that this group received the same type of transplant without the addition of T cells, whereas this group received the same transplant but with immune cells administered a month later. And you can see that it makes a huge difference. I'm showing you a picture of one of my patients. Actually, he's one of those patients in the first study that I showed you. This young individual, Eric, had acute leukemia. He was going around with leukemia cells. He'd had multiple cycles of chemotherapy that were actually unable to control his leukemia. He was going around with leukemia cells, and normally we would say he's chemo refractory and not do anything. But because we had this protocol, he was eligible. We put him on the protocol, and the patient could have a transplant from his father. Before this protocol, we would not have done this strategy. His father was only half match. Actually, he was 4 out of 6 antigen matched. And interestingly, this patient reached a complete remission for the first time after the transplant. Not only that, 9 years after, still no acute leukemia. So it's not a study, but it tells me it's extremely compelling, and to me it's almost as convincing as all the curves that I've shown you, that the immune system is able to do what other strategies are not able to do. And one of the obstacles actually associated with this strategy, the fact that individuals are half-matched, could be an obstacle in terms of graft-versus-host disease. But actually we think it's an asset for fighting the leukemia, because there's a possibility of having an immune stimulus against these cells. By the way, he has agreed to have his picture shown, by the way, and I see him on a regular basis. And actually his picture is part of a book that another of our patients has prepared with pictures of patients to give hope And I think that's what we need to give these patients, hope that there is survival and there is a possibility of going back to a normal life afterwards. Okay, I'm going to shift gears and I'll tell you about other things. Like I think the example I just gave you is pretty compelling, that the immune system can generate substantial improvements into these patients. And what I've shown you is that we have cells that are able to generate anti-leukemia and anti-infection. Because the curve below that I showed you, the patients that died, and this is a hard outcome, you can't cheat on alive or dead, the difference is really infection and leukemia. But we won't accept this situation. We need to continue and find new strategies. So another strategy that we've been developing is try to even be as selective as possible. And what I will be talking to you about is targeting what we call MIA, which is minor histocompatibility antigens. And what we have been talking about is that with stem cell transplant, or what we call conventional allogeneic hematopoietic cell therapy, or cell transplant, or stem cell transplant, what we're doing is fairly rudimentary and really toxic, because we're taking normally stem cells and just administering them to patients, hoping that among the cells that we're giving, some of them are going to recognize the leukemia. What we would like is to really develop cells that have the ability to specifically recognize the cancer cells. How do we do that? Well, interestingly, our immune system has evolved a fairly sophisticated system for tracking cells. So if you have our immune system, and here is a killer T cell, a cell present in the immune system, it has on its surface what we call a TCR, or a T cell receptor. Now this T cell receptor is able to recognize cells that are presenting self-peptide. So the peptide is a portion of protein that's expressed on this molecule, and we have, all of us, we have these molecules on the surface of all of our cells. And in particular, what we want is to be able to identify the leukemia cell and the other cancer cells that harbor peptides such as these, and enabling them to be recognized by T cells. Is it possible to do that? Well, first, what is a minor histocompatibility antigens? Well, they come from our machinery. The cells are actually making proteins, and this is part of their function on a daily basis. And the proteins are eventually cleaved. They go through a proteasome, and then they are transported to the surface of the cell. And this is how our immune system recognizes our cells versus cells from somebody else. They recognize the combination of the peptide, small portion of protein, attached to this molecule, which is called the MHC, major histocompatibility complex. The T cell is highly sophisticated and is really able to specifically recognize these cells. And what we have been working on is identifying those peptides that are specifically expressed on leukemia cells. I was telling you that the immune system is fairly sophisticated. Well, it's so sophisticated that the T cells of an individual are able to recognize peptides that differ from one person to another by only a single amino acid, so the smallest part of a protein. So the T cells are able to recognize this, whereas, for example, our own T cells are not reacting to our own cells. But if you mutate this peptide at a single site, then the cells will be able to be recognized. So there have been studies done with my colleague, Dr. Claude Perrault, who's actually at the origin of this and has been studying these peptides for many, many years. It was possible to develop a strategy where in mice it was possible to identify peptides, this one called this one B6-DOM, that was the name that was given to this peptide. It was possible to generate these cells against this antigen, and when we had leukemia injected into mice that harbored this protein, the mice all died. But when we administered the same mice, we administered cells reactive to this peptide, they all survived. And interestingly, was it not only good for leukemia, but similarly for a solid tumor like a melanoma, which you know how aggressive it is, we had the same type of reactivity. So that's highly encouraging. In humans, it had not been done. So the strategy here was to take some cells from various individuals and try to identify the changes in amino acid between two individuals, allowing recognition of the peptide. And how that was done using cells, immortalizing them, and then using what we call mass spectrometry to study the small peptides on the surface of the cell. The sequence was then compared to a data bank to try to identify the single amino acid differences between two individuals with exactly the same peptides, and then generate a large bank of candidates. So there were actually 15,000 such peptides that were identified. Out of these, there were some that are highly infrequent. We didn't want to find, peptides that are only present in a very small proportion of the patients. So if you look in large numbers, so those that are expressed more frequently, this brought down the number to close to 500. But then what was sought is peptides that would be preferentially expressed on cancer cell and not expressed on other organs, for example, like the skin or other tissues. And this restricted the numbers to 98. And this is now what we have, 98 new minor histocompatibility antigens that can be targeted on leukemia cells. So with these, we have immunized cells in the laboratory. So we've taken donor cells, pulsed them on the surface of dendritic cells. Now, dendritic cells are the cells that are what we call professional antigen-presenting cell. Their job is to show off the antigen to the cell. And we were able to derive a protocol that allows us to produce T cells, so immune cells that are able to recognize these peptides. And although it takes a fairly long time, we are able in the end to generate cells that recognize leukemia and are able to kill them. With a grant from these agencies that you see listed here, and the peptides identified by Claude Perrault and the team, Sébastien Lemieux and Pierre Thibault and others, using bioinformatics, as I was telling you, mass spectrometry, etc. It was possible, and these 98 minor histocompatibility antigens, what we are now doing is taking patients that have relapsed after a transplant to test the strategy. So we are taking patients who have had a transplant and where the leukemia comes back. We take the donor cells, we immunize them in the lab, so we add the peptides and therefore grow them according to the protocol I just showed you, and generate T cells with the ability to recognize these minor histocompatibility antigens. And then we take these cells and we re-administer them into the patient in order to clean up the leukemia. This trial has just started. Already results are quite interesting, but we're only at the beginning. But I think this is one of the strategies that, again, is extremely interesting and promising. But in order to get there, we had to be a whole team of people working on this. I didn't mention Jean-Sébastien Delille, who was key in setting up all the culture conditions, and the whole team, actually a large number of people, are involved in this. Why am I presenting this to you with such enthusiasm? You kind of wonder. Well, there's a reason. We know that T cells are efficient. I showed you some examples of haplotransplant. Let me show you another example of antiviral T cells. So we had a patient who was actually a post-transplant patient that developed lymphoma in his lung. So what you see is not normal. These are actually infiltration by tumor cells. And this is what we call a B-cell lymphoma. And it's a post-transplant lymphoproliferative disease. Very long name to say immune system is not able to tackle the virally infected cells. But it's a tumor. It's from a viral infection, but it really is to the stage of a tumor. This patient got chemotherapy, didn't respond. We gave him antibodies. Regular treatment is antibodies. Multiple cycles of antibodies, no response. Patient was basically had no therapeutic option. So we took cells from his donor, showed them the antigens, the peptides presented by the virus, generated cells in 10 days, and re-injected them into the patient. And here you can see the result. Really amazing. And this is 3.5 months, but I can tell you that I saw the patient on Tuesday. His CT scan is now normal. He had sinuses actually full of lymphoma, completely cleared. The ENT surgeon, I wanted him to clean it up. He said, there's no way, I have to basically remove everything. So we didn't do anything, we waited. He's cleared up everything just simply with the addition of cells. And this is not the only one. We're not the only ones doing this. People at Baylor are developing this. There are people in Europe working on this. So clearly, there is a big future for cells actually to not only fight cancer, but also to fight infections. And here in this case, this was cancer. And it's interesting because this patient sometimes has a rise on the antigens found by the virus. And his immune system is able to kick back. We then find a surge of the cells coming back and going away when the infection is under control. Pretty amazing. Now all of this you could see is all tedious. At the current time, we're working and we're manipulating the cells, and we're doing this in a first generation. And when you're talking to people are saying, yeah, well, cell therapy is very interesting, but it's very labor intensive, hard to do. How are we going to make this into a process that can be used efficiently in patients and be able to treat hundreds, if not thousands of patients? Well, right now we're doing this all manually in suits like this in order not to contaminate the grafts, because we culture them sometimes up to a month and a half. So that's too long. We could use some robotics. It's going to help, but clearly not the option in the long term. But what I can tell you is that there are various strategies, including automated bioreactors, for example, that are being generated, that allow translation, that are acceptable on a regulatory basis for regulatory agencies like Health Canada, that are scalable, that are economical, and that allow everything, including quality monitoring. So there is a future for these strategy, but we can't stay at this stage for too long. I want to tell you another opportunity. CAR technology. So it's not the CAR that you think, but CAR stands for chimeric antigen receptor. So T cells, I told you, have the ability to recognize cancer cells. That's normal cell. But they don't always do that. The proof is that we have cancer. So the idea here is to take a gene and introduce that into these T cells in order to increase their ability to recognize the cancer. And what is this gene? Actually, this gene corresponds to a new receptor that has the ability to recognize antigens found on the surface of cancer cells. Is this the future? No, it's the present. Interestingly, there are a number of targets that have already been identified more than 20 years ago. Antigens like CD19 and CD20. So CD19 is expressed on cells, on peripheral blood cells. And if you take here is a stem cell, and here is a very mature antibody-producing immune cells. Well, all the cells in the middle are all expressing CD19. Even those here are expressing CD20. And this is very interesting because it's not only present on B cell, but it's present on the cancer cell. And you can see here various leukemias, chronic lymphocytic leukemia, lymphoma, and other cancers. And what's interesting with this antigen is although you eliminate those B cells, they can be reproduced from hematopoietic stem cell that can replace those cells that are eliminated. How does it work? It has this CAR technology as an external domain, so it has the T cell produce this new molecule, which has one part with the ability to recognize the antigen. And what is really new is that inside there are some what we call co-stimulatory signals. So signals that tell the cell, kill this individual. People have been working on this for more than 20 years. They had put this part alone. If you only put this part on the surface of the T cells, it's interesting, it binds to the cancer, but it doesn't kill efficiently. But since the introduction of these stimulatory domains, this molecule suddenly renders the T cells highly active and makes them really potent killers. How potent? Well, here is a study, 48 pediatric patients with relapse or refractory acute lymphoblastic leukemia. 94% in complete remission with this molecule. Pretty impressive, above what can be achieved with chemotherapy. 6-month survival, 81%. And you can see here the survival curve. This is totally amazing for this type of patient population. This is so amazing that this strategy has been approved by the US FDA without a randomized trial. So it hasn't even been compared to another treatment. People have seen these results, they say we cannot achieve that regularly, and therefore it is now on the market. Interestingly, for example, the CD19 antigen I told you about is present on a number of cancer cells. We were talking B-cell acute leukemia, lymphoma, chronic lymphocytic leukemia, and various types of cancers of the lymph nodes, potentially even on precursors for multiple myeloma, although this is currently debated. So this is extremely promising and under development in all of these diseases. Now the problem is that we need to find other targets. CD19 is interesting, CD20 is there, but not all cancers. So right now there's a race to try to identify other antigens that, like CD19, could be targeted and found on cancer cell and link them to a structure that we've just been talking about. Other strategies in immune function? You may have heard of checkpoint blockade. We recently discovered that our T cells, actually, which I told you, recognize tumor cells through the T cell receptor. Now you know everything about this. So you have the T cell receptor, the MHC molecule, the antigen, the peptide expressed here. But what we hadn't realized is that there are other molecules that are there that actually put a hold on the activity of the cells. What they are is actually they're a foot on the brake. So when the T cell recognizes the cancer, it binds to the tumor, but at the same time, by linking what this receptor PD-1 links with its ligand PD-L1, and then what it says, it tells the T cells, don't be too active, don't kill too much. What recently has been done is actually have the ability to dissociate these two parts, and therefore it removes the brake and allows these cells to be even more cytotoxic. So cytotoxic that this generated the best results that we've seen in cancer in years. This is one of those brakes. This is CTLA-4, an antibody that was known. Adding a PD-1 inhibitor, as you can see, more than doubles the efficacy of this treatment in a very deadly condition, metastatic melanoma. Lung cancer. As you can see, standard therapy, we don't have these great results. You add this molecule, nivolumab, and you can see here, actually even just as a comparator, it has an improved activity, and when you start combining that with other agents, you get even better results. So this is drastically changing the scenery of treatment in the solid organ area. Are we seeing the end of this? Well, it's only the beginning. I was telling you about CTLA-4, which is the other molecule I was just telling you about, and PD-1. But here you can see there are a number of inhibitory receptors that can be blocked, and people are working on this. There's also a number of receptors that can be activators, and for which molecules that stimulate can be generated. So there's actually a big effort internationally to work on these various molecules to increase the efficacy of T cells to fight myeloma and other cancers. And interestingly, we even increased our understanding of this process, and recently an investigator at our institute, Christopher Rudd, who has recently moved from Cambridge to Montreal, to Maisonneuve-Rosemont Hospital, so he's now a Canadian researcher, has identified in a very prestigious journal a new pathway. So glycogen synthase kinase 3 inactivation actually drives the activity here of TBET, which actually leads to downregulation of PD-1 and therefore increases the activity of the T cells. And you can see here that if you have a healthy mouse in which you administer cancer cells, so B16 cancer, and you have no treatment, the black dots represent cancer growing in the lungs. When you add an agent that is a GSK3 stimulator, you see a decrease in these black dots. If you use anti-PD-1, you get similar activity, and if you combine both agents, you have even better activity. So actually, this is even a new pathway that can be targeted and seems to have extremely high potency in driving T cells against cancer. There are other strategies that are not necessarily directly aimed at eliminating cancer, but are also helping us in our ability to fight cancer. And I'm giving you one example, and that's our ability to grow stem cells. And I told you, so you all know about how to produce stem cells, why they are needed. But the problem is that in many instances, when we take patient cells, patient has had lots of chemotherapy, we want to do autologous transplant, there's not enough T cells, or we have a compatible donor but we don't have enough cells, or we have a umbilical cord blood, and it's highly useful for transplanting a 6-pound neonate, but it's not very useful to transplant somebody that's 200 pounds. So if you want enough umbilical cord blood stem cells to transplant adults, in many instances you don't have a good choice of cords that provide sufficient numbers of cells. So actually, Guy Sauvageau, Canadian researcher at the EHIC in Montreal, has identified a molecule that allows the expansion of the stem cells. So it allows us to grow stem cells in the lab, and that these cells are able to generate hematopoietic cells so that the stem cells can be administered to patients in order to replace their bone marrow. And this is currently under study. A number of patients have already been transplanted, patients engrafted, and the patients are doing extremely well. So it's helping us in our ability to fight cancer and to deliver stem cell transplant. So we will see more and more of this as this goes on. How am I doing with time?
Are you close to?
Yeah, I'm going to
leave time for questions.
Yes, sorry. Maybe I want to end on saying that there are a number of applications for using stem cells. Heart diseases, those stem cells to help repair the heart. And there are a number of trials, either injecting these cells into vessels, directly into the heart, or during surgery into the area that is not functioning well. We now have strategies to isolate cells that we want using magnets, for example, and beads, antibodies attached to beads. We have the ability to modify cells, I was telling you, with gene therapy, and that can be used also to help regenerate organs. There are clinical trials using that for patients who've had a myocardial infarct, where a gene is introduced that helps keep the vessels open and helps cells regenerate the tissue. There are cells for hypertension, for example, run also by Duncan Stewart in Ottawa, and actually trials conducted throughout Canada. There are ways of generating skin, and there's a lab in Quebec City at the LOEX that are generating biolamellar skin, which is enabling patients that are severe burn patients to survive their episode, and that's done on a regular basis. Strategies to replace eyes, to corneas that are opaque and allowing them to regain vision. People working on scaffolds to help the cells grow. People developing mesenchymal stromal cells for arthritis, which have various properties, replacement, but also removing the inflammation associated with arthritis. And there are trials ongoing right now. Actually, this trial is finished and quite interesting and favorable. There's possibility of generating these cells from various sources, including the umbilical cord itself, not the cord cells, but through the cord we can isolate mesenchymal stromal cells. And there's the team, and obviously a lot of people here in Vancouver that I didn't touch on their work. I would be, I could not deal with such elegant science that is done by Keith and the others, Tim and the others, Jamie, Megan Levings, Keith Humphreys, Connie Ease, Fabio Rossi, etc. There's a team in Edmonton that I didn't touch upon, but that's also working closely, collaboratively with the people here and others across the country that are developing a variety of strategies for using cells to tackle immunologic and degenerative diseases. And I want to end on the fact that we have created a network that Tim was alluding to, which harbors not only includes cell manufacturing centers, but it has legal platform that's headed by Bartek Knoppers. We have developed cell therapy centers across the country. I'm just showing you the one in Montreal here, which is probably the most developed, 4-story building, totally dedicated to cell therapy. And that's what people look like when they work in these environments. So you don't want them to contaminate their grafts, quality control, freezing of the samples, and all electromechanics behind it to ensure that the buildings are working appropriately. We've developed Center for Commercialization of Cancer to help translate these exciting findings from our great researchers into clinically applicable options. And develop biomarker unit, manufacturing unit, clinical research unit, and innovation units. Finally, all together throughout Canada, we are sharing best practices to CellCan and are developing, as you can see, protocols for almost any organ. And so if you want to learn more, I would invite you to go to our website, which is www.cellcan.com. And as I was saying earlier, if you don't remember, you type ilovestemcells.ca and you'll get there, or j'aime les cellules souches en français, and you'll get there. And it has news. We're on Facebook, we're on Twitter, and we'd like to showcase your great science, and we need you to go to the site and inform us and let us know how to work. There are a number of information booklets that are available. You just go to the website. There's even an app that tells you what studies are ongoing in various studies, and we're going to have a 2.0 version of this app, which is actually pretty unique worldwide. And with that, I would like to end on thanking all the collaborators, the people that are involved in this, the cell manufacturing team, the research labs, all our funders. Thank you very much for your attention.
Thanks very much. Really exciting to see the potential of cells. Okay, we've got a few minutes for questions. Yes?
Thank you for your very elegant talk. I'd like to ask you a question about tumor cell burden. Is tumor cell burden limiting to the effects that you can obtain? Because there's certain, as you told us, a certain number of cells that can actually, T cells, stem cells, that can be given to patients. But if the tumor cell burden is high, is there some kind of an amplification factor in vivo that can help them take care of more tumor cells that they can encounter? Like if one T cell will kill one tumor cell, or can one T cell kill 100 tumor cells? So tell us about the role of tumor cell burden.
Yeah, that's a very interesting and difficult question. It is clear that there is a limit to what the immune system can tackle. And I think that up to now, when we were doing regular transplant, the activity was most potent when there were few cells that were left. For example, if you didn't give chemotherapy and radiation, you wouldn't be able to do that. We're amplifying this activity. So clearly the tumor bulk that is amenable for the immune system to be eliminated it has increased. So we have increased our efficiency by boosting the immune system. However, clearly the tumor mass is a determining factor. And we have given cells, for example, to patients with highly active disease, and I think that by themselves it has not been sufficient. Do we have a real good number, like what is the minimum number or the maximum number that's treatable? We don't know, and it's probably very different from one individual to another. If we're talking haplotransplant, we have, and there is no good study. I can just give you some examples where patients actually have, for example, 50% leukemia. Give them some cells that are half-matched, haploidentical, so immunologically very different. Well, a single injection of cells was able to induce a complete remission in these patients. So this has been observed in many patients, and this has been done after transplant, and the effect is probably greater after haploidentical transplant than after MHC matched transplant, for example. In other instances, this has not worked. So clearly, I think that there is a need for decreasing the tumor load, not only as a decrease of the tumor load, but potentially also through other activities, such as killing the cell, and therefore enabling release of parts of the cells which can then be recognized by the immune system. So allowing dendritic cells and others to grab the antigens and present them appropriately to the immune. Yes. So we think that the chemotherapy not only has an effect, for example, on decreasing the tumor load, but also on increasing the immunogenicity and the ability of the immune system to recognize the cells. And same way, it has been shown that if you make room in these individuals, if you lymphodeplete, if you eliminate the white blood cells from the individual before administering lymphocytes, the activity is better. So there are a number of factors that are important. It's not a single one, but all of those are important. But what we are seeing is that the bulk of cells that can be tackled by immune cells is going up, and that's highly, highly encouraging.
Other questions? Yes?
This was fascinating. Thank you so much. I was wondering, I read about TGF-beta coming out of platelets and GARP TGF-beta axis being important to inhibit the T cells and maybe even CAR T cells. Could you expand on your opinion whether this has importance or?
I'm not an expert on TGF-beta, and actually one of my colleagues is, so he would answer much better than I would. Interestingly, yes, I think TGF-beta is one of the suppressor molecules that we think is playing a deleterious effect. But I was reading recently that it could also be the opposite, that it could be beneficial in some instances in allowing other mechanisms to be active. So I think it's not clear. It's not a single molecule or a single effector pathway that is going to be involved. But I'm unfortunately not an expert enough to answer your question appropriately.
Question over here. Yes.
Hi, thank you for the very interesting talk. I just had a question about, so you have a few slides about checkpoint inhibitors. So I know that right now some biomarkers that we're using are, for example, like the PD-1 or PD-L1 through immunohistochemistry. I don't know, what are your thoughts about the development of other types of biomarkers for checkpoint inhibitors?
Well, clearly we're going to need to be able to measure that both on the tumor, as you know, and on the effector cells. I would compare that as to NK cells. When we started looking at NK cells, it was KIR mismatching, and we were looking at one or two KIRs, killer inhibitory receptors. And now we have a whole range of molecules that we're looking at to really measure the ability of NK cells to kill cancer. So I would say that in this setting, it's most likely that we will need to assess the whole spectrum of the cells that are expressing this. But clearly, measuring levels of PD-1, PD-L1 would be useful. It's just, I think we're only seeing the beginning of this. But clearly, biomarkers are going to be a tool, and it won't be the answer to everything, because there will be people expressing or not expressing PD-L1 that are still remain refractory to treatment with anti-PD-L1 blockage. So that is currently being found. So it's not a simple answer. It's not a simple biomarker answer, but it's part of the answer. I think we should do that, definitely.
Can I get you to comment on the cost of the CAR T cell therapy? You mentioned that this is exciting, it's been improved. Tell them how much it costs to treat a patient with this.
Yeah, well, it will all depend on, we don't know exactly how it costs, but somewhere between $400,000 and $1 million. There are some per patient. The argument being, well, it's a one-time thing, you give it once, the patient is cured, and therefore if you were to treat him for years and years, it would cost you more than having a single treatment. But you imagine the number of patients with cancer, we can't afford to say that.
So I wanted to provoke you to comment on what is it going to take to make the cost come down and make it more reasonable, and what's kind of the timeline to making this more mainstream?
Yeah, and actually the companies are very smart. Some of them are really thinking that they will only charge you if the treatment works. So confident that it's going to work that they will not pay. But you're right. I think, one, the cost is going to be high. If you think about it, the cost of a stem cell transplant is about $100,000. I'm making a round number here. It can go from 80,000 to 120 to 200 in some cases. So it's fairly expensive treatment. However, right now the big cost is really the cell manufacturing. Actually, there are two costs. One is the cell manufacturing, and the other is the handling of the patient. Because they have been experiencing a number of side effects. Some of them have gone to intensive care unit. They have gone to what we call cytokine release syndrome. Before, we didn't know how to treat it. Now we know how to predict, not to predict, but how to treat it. There's still some side effects like CNS neurologic syndromes that have been described that are currently observed with this. We don't know whether it's the expression of the antigen on the surface of neurologic cell or whether it is with the CAR going into the brain. So it's not devoid of effects. However, the activity is so overwhelming that it's there to stay. So coming back to your question, it can be I think we're only starting. The way it was done is it was developed in numerous labs, mainly Carl June and Michel Sadelin and others, and it's been done manually. So we have people and we have small bioreactors, but it's not an automated process.
But it will become.
But clearly it's going to become automated robotics. Time is going to be decreased. Jamie is going to find a way to increase our efficiency and decrease the need for gene delivery. So I think people are right now, it's incredible. The whole planet is working on improving these strategies. And to me, the cost is going to remain high for the first patients, but after a while it's going to decrease. I think the obstacle is going to be different. The obstacle is going to be finding the right target, because the technology is great. CD19 is the perfect target, similarly to what BCR-ABL in chronic myelogenous was when we developed tyrosine kinase inhibitor, Gleevec, which has taken the world by surprise. And now has rendered leukemia a chronic disease. You give a pill and you cure chronic leukemia. But is it going to be a blockbuster in the other cancers? It will only be true if we identify the right antigens. And we've had problems identifying antigens that are specifically expressed on tumor cells.
Well, you've given us hope that we're going to get there soon, so thank you very much. And just before we close, I just want to mention that this is part of a series of talks. If you've enjoyed this one and you want to learn more about different areas of stem cells and regenerative medicine, we invite you to come back to hear these. And I would like to acknowledge the Killam Fund at UBC for supporting this lecture series. So, Dr. Roy, that was very exciting to hear this work firsthand. It gives us a lot of hope that a cure for cancer is going to be widely available for people soon and hopefully affordable. So thank you for sharing that exciting development with us.
Thank you.